The title of our talk tonight is Genetic Testing of Honeybees for UK Beekeepers, and I'm delighted to introduce Dr. Mark Barnett, who is our first speaker. Mark is a Scottish Expert Beemaster and looks after the apiary at Edinburgh University. So it's good to have both a beekeeper and a bee scientist talking to us tonight. So over to you, Mark. Thank you very much.
Thanks very much, Bob. I'll just share my screen. Can you all see that? Yeah?
Yeah, I think everybody's muted, Mark.
Yeah. Or somebody. Okay. Yeah, well, thank you very much, and thanks very much for inviting us to speak and tell you about our community interest company, Beebytes, which is the main subject of tonight. I'm going to start off by giving you basically a review of honeybee research and activity at the Roslin Institute. I actually manage the bees at the Easter Bush campus apiary, which is actually out south of Edinburgh, 8 miles south of Edinburgh, where the Roslin Institute is situated as part of the veterinary school. And Matthew actually manages the University of Edinburgh apiary, which is situated in the middle of town. Okay, so yeah, that's the Roslin Institute, and basically it's made many contributions to animal sciences, especially in the area of livestock improvement and welfare. So my background is not in honeybee research. My background is in developmental biology, and that's studying how embryos develop and how genes pattern embryos. So I came to bee research through beekeeping, and mainly through I've been beekeeping since 2010, and so three years into my beekeeping, there was this citizen science project that was run by Science and Advice for Scottish Agriculture, which is like our version of the NBU, and the University of Dundee and the Scottish Beekeepers Association. And here we see Fiona Hyatt outside the laboratories at Sasa with Scottish beekeepers Alan Reich and John Durkax. And basically, it was a citizen science project, and we were going to map diseases throughout Scotland. And this was thought to be, basically, this project impressed the Biotechnology and Biology Science Research Council, which is one of the major funders of the Roslin Institute. And they really thought this was a landmark in citizen science. And the reason for that was, the beekeepers basically collected the samples. They then screened the samples, so there's a crush in a bio-Reba bag, and then identified whether they had nosema by microscopy locally. And then if they got positive samples, then these were sent in by post to 9 citizen science beekeepers, of which I was one. And they were trained up in the polymerase chain reaction, so they could detect both species of nosema by PCR. And this shows the result of that project. We've got the distribution of Nosema serrani and Nosema apis in Scotland, and you can see Nosema serrani in red. We've detected quite a lot of Nosema serrani, but not very much, interestingly, not much Nosema apis. And actually the West Coast, there wasn't much Nosema at all detected. And I think what really impressed me about this project was that, what a lot of work people can do when we work together. And not only did we do both nosemas in Scotland, we also did deformed wing virus as well. And you can see the positive deformed wing viruses there shown in orange, positive apiaries. Not surprisingly, out to the West Coast, where we've still got a few Varroa-free areas. We've got quite a few negatives. I think the positive one on mull I think there wasn't any Varroa on mull at that time, and we think this isn't Varroa. This is bees get deformed used to get deformed wing virus anyway before Varroa got involved, although it wasn't as much of a problem then. Okay, so while we were doing this citizen science project, there was a lot of conversation in the lab and talking about PCR, beekeeping, etc., science, a lot of interaction. And Alan Rieck, who was at the time the SBA education convener before he was the SBA president, he asked me about, do you think we could get some DNA regions that determine, we could detect temper, imagine using this in urban beekeeping. So I had a conversation, I was working for Professor Tom Freeman at the Institute at the time, and we thought about it and thought, well, if you're going to go looking for the DNA that's behind a trait, you don't want to go looking for temper, because as we beekeepers know, temper varies through the day, it varies through the year, it varies with what the bees are on, and it varies with which beekeeper is going through the bees. So basically, if you're going to go and look for some DNA, you want to get a measurement that's really sort of firm measurement. So we thought that's not a good idea at this time, but it did focus our mind on the fact that the knowledge of honeybee genetics was lagging way behind the other farm animals that we work at the Roslin Institute. And so this led us to propose that we would sequence the UK population. We haven't sequenced the UK population, but we did manage to do a pilot. And basically we sampled 19 colonies across the UK. We isolated, and each sample was DNA from 16 workers from each colony. And then we sequenced them by using whole genome sequencing, we're using Illumina technology. And just to tell you something about those samples, these 4 samples here were down from England, and they came to us as good examples of Apis mellifera, the native bee. Whereas the Scottish samples, apart from obviously the good example of known to be of Apis mellifera on Colonsey, I've lost my mouth. Oh, right. The other samples from Scotland were a random sample basically that we took, and we also took a Buckfast sample and a Carniolan sample from recently imported bees into Scotland. And so we sequenced them, and this graph is basically a similarity network, and it's basically comparing the genetics of all those colonies. And so if you look over each of these spheres represents a colony that we've sequenced, and we're comparing the genetic variation. And basically the thicker the lines, the more similar the samples are. And you can see here for Colonsey, which is a good example of the native bee, we've done this at, it says times 50 and times 25, and that just means that we did this sample twice, and we sampled it for if it says times 50, then each base was sampled an average of 50 times in the genome, and 25 times. Each base was sampled 25 times. And the thing to note from this is we've got this cluster here of these, which were essentially samples taken randomly from Scotland, which are very closely related to the Colonsey bee. Whereas the samples of AMM that we took from England, which were given to us as good examples, are less related. We've got our Carniolan and Buckfast over here, that they're not connected to the graph, so that means we could place them anywhere. However, if we were to lower the similarity for them to be connected to the graph, then we would get a graph like this. It's not quite as pretty, but it does tell you where those the Buckfast and the Carniolans are sitting. So basically, these bees here are closer to the, have got more C-lineage in them, and these bees here have got more M-lineage. When you sequence the DNA of a bee, you don't just get the DNA of the bee, you can get basically all the organisms that are within it, its associated microbiome. So you can get the bacteria that are in the gut, and then you can also detect its pathogen load. And this work was done by a colleague of mine at the Institute, Dr. Tim Regan. And I'm not going to explain exactly how we did this. If anyone's interested, I'm happy to, if you email me, I can tell you how we did this. But basically, he was able to identify in the DNA from those colonies all of these organisms. So you can see we've managed to detect Nosema apis, Nosema ceranae. We've managed to detect chalk brood. We've got a trypanosome, Lotmeria passim, and we've basically detected all the common bacteria that are found in the honeybee gut: Lactobacillus, Bartonella, Gilliamella, Snodgrassella, Fischella. Another project that we got involved in, and this was a collaboration with a scientist in Portugal, Alice Pinto, and this was a low-cost genotyping platform which was to analyse C-lineage introgression in M-lineage honeybees. And basically what Alice had done is she'd taken the available knowledge at the time, and we've got much more knowledge of bee genetics now than when she did this, and she selected small nuclear polymorphisms, and she selected the ones that were most different between M-lineage and C-lineage. And we submitted 96 samples from the UK, including some samples from the B4 project in England. So, and these were analysed for admixture, and admixture is the result of breeding between two or more previously isolated population within a species. It's basically, you do it on a computer program to analyse the proportion of the genetic background. When you run the program will assume different numbers of genetic backgrounds, whether there's 2, 3, 4, and basically it'll work out which one is the most significant. So in terms of this genotyping experiment, it's 2. And basically you can see here, this bee here, if there's 100% probability that this bee is M-lineage ancestry, and then this bee over here, 100% that it's C-lineage ancestry. And if you put all those columns together and you order them with the M-lineage to the left, the most M-lineage proportion to the left, then you can make a graph like this, which was the one that appeared in the paper. And the thing to note from this is that yes, it confirms what we found before, and we seem to have more good examples of the native honeybee in Scotland than from our samples we took in England. The good examples we did get from England were from Cornwall, from the B4 project. And that's also Colonsey bees. And then also the thing to note is that in France, if you look at this, then basically the protected areas in France don't seem to be working as well. Another piece of work we've done is basically working towards a gene expression atlas of the honeybee. And basically knowledge of the honeybee genome is lacking way behind other livestock species that we study at the Institute. And if you look through the honeybee genome, you'll find many genes are of unknown function, and most of them have not been given proper gene names. So our aim is to create a gene expression atlas for the honeybee. And ideally, we would analyse expression of all genes in all sexes, castes, and all stages of development. And what use would that be? Well, if you wanted to look, for example, if a scientist wanted to look for the DNA that determines wing morphology, one of the things he could do was he could start by looking at all the genes expressed in the developing wing. So this is what we've done a pilot towards this, and this is what we've done. So basically we took some pre-pupa and we took some pupa at day 15. These are workers. We divided them into single cells. We then sequenced their mRNA and then analysed. And this shows the result of those. So each of these spheres in this graph is one of the cells that we sequenced, and we've compared them in the graph so that the similarity by similarity of gene expression. So these cells here expressing similar genes, and obviously the ones over here, these are expressing much different genes to the ones over here. And we can see that we got more cells from the pupa at day 15, and the pre-pupa being shown in green. And basically, we were able to cluster these cells, and we were able to identify 63 different clusters, so which might represent 63 different types of cells. So the next thing I had to do, and I spent most of lockdown doing this, was to find out what these cells were. And it was basically the sort of lack of knowledge on in the honeybee made it a really difficult job. But this is one of the tools that I used to identify what cells were what. And so basically, if we've got a gene, and we take, if you look at this graph here that says C2 neuronal, basically on the x-axis, we've basically got cells. These are the cells that we got from the pre-pupa and the cells we got from the pupa. We can basically look at the expression of a gene, and so for this gene, it would be expressed high in these cells, and then low in these, and high in these, and then low in these. And then we know that this is the brain or nervous or part of the nervous system, because we know there are known neural genes that are expressed in this cluster, which is shown here. And so we can basically do this with all these different expression, and as you can see, we've able to identify cells from the midgut there. And so that was the final result. It probably doesn't look that impressive for how much work went into it, but you can see that we've identified cells from the brain, the eye, cuticle, muscle, Malpighian tubule, hemocytes, midgut, and glia. And I think really the good thing about this is the proof of principle that we can do this on a single cell basis in a developing bee. So just to summarize what I've talked about, we think DNA sequencing is becoming a more cost-effective platform for analysing genetics of honeybees, and it's getting less and less expensive. We've shown that Scotland might be a good place for conservation of the native honeybee, maybe because of the climate, maybe because of its geography. And the technology we showed for analysing the microbiome and the pathogens, well, you could imagine this could be developed, and you might use that for screening, for example, imports into the country for bee health. And then with the Atlas work, we've basically shown the potential there to greatly improve current understanding of honeybee biology through single cell sequencing. I'll just briefly mention activity at the Roslin Institute to do with beekeeping. We established an apiary mainly for research activities, but it's been expanded now to look after staff and students. Staff and students can now keep bees on campus in their lunch times. Sometimes they even make honey. We've found that honeybees and beekeeping has been a great way to engage the public about science. I even had to go to a high security prison. Funnily enough, they wouldn't let me take a hive tool in. If you Google hive tool, it says a sharpened metal instrument used for beekeeping. So I think they let me take that brood box in, and that was it. I think that's one of those learning hives. And also we've been, at the Institute, we've got the Easterbush Science Outreach Centre, and we've been doing various systems and science projects there in association with the Scottish Beekeeping Association. Other exciting news from Roslin is there's a PhD started in September in Gregor Gorjank's lab, Laura Strachan, and she's doing a project on design of breeding programs to improve honeybee health and production. So we're very excited about that project. And the subject of tonight is we've recently formed a community interest company called Beebytes Analytics CIC with my colleagues David Wragg and Matthew Richardson. I'm going to let them tell you all about that, and it's based at the Roslin Innovation Centre, which is next to the Roslin Institute. And that's just all the people that have been involved in that work. And last but not least, the citizen science from the citizen science project I spoke about at the beginning. Thanks very much.
Thank you, Mark. Is it David's going to carry on now?
Yeah, I'll just share my screen. Just minimize that. Okay. So I'm just going to go into some of the points that Mark mentioned in a little more detail. My background is in population genomics, so I've worked on chickens, camels, sheep, humans, honeybees. I do have a track record in publishing on honeybees as well, but I'm the odd one out here because I'm not actually a beekeeper. So I'll just outline whole genome sequencing in case anyone's not familiar with how it works. Go into a little bit about what a native bee might be, talk about the drone haplotype we've been working on, the HapMap, what a graph genome is, and then discuss a bit more on the iPlex mass array that Alice Pinto developed, as Mark mentioned. Go into a little bit of detail about guts, the microbiome, and why they matter, and also on DNA metabarcoding. So with respect to whole genome sequencing, a brief outline of the process is that the DNA is extracted from the tissue, and this is fragmented for what they call library preparation. And that involves the addition of sequencing adapters to those DNA fragments. So when we sequence those, these typically give us what we call pair-end reads. So that's a pair of reads that are 150 bases long. And it generates millions of these sequencing reads per sample. Now, the honeybee genome itself is 235-ish megabases in length, and we have to align all of these paired reads to a reference genome. So for the honeybee, we have two options there. There is the main reference called the DH4. This is a strain from the US, believed to be derived from Ligusca, so the Italian bee. Another option is to align to a reference of Apis mellifera mellifera, the black bee, which was developed by INRA, so that's the French National Institute for Agricultural Research. And that bee is from the Ile de Wesson, which is a little island off the coast of Brittany, so much like Colonsay is to Scotland. So we align all of these reads to this, what we call linear reference genome, because the reference just represents a single individual rather than the whole species. And we identify genetic variants that we term single nucleotide polymorphisms by comparing the nucleotide base we observe in the sequence data to that of the reference genome. So these SNPs, as we call them, we typically find about 1 million per individual bee that ends up being sequenced. So just to touch on what Mark mentioned about Tim's work on identifying all those various microbiota when they did the whole genome sequencing of bees. When they extracted DNA from the abdomen of the bee, that also allowed them to capture the bacteria in that abdomen. Now, obviously, when you align these sequence reads to the reference genome, that doesn't represent the bacteria. So it's a case of taking all of the reads that don't align to that reference, sort of de novo assembling them, a bit like a jigsaw, and then cross-referencing those, what we call contigs, against the reference database to see if they resemble any known organisms, such as the bacteria. And that's essentially how they constructed that graph. So once we have all these, the SNPs identified, all these variants, we can do a number of things. And most commonly, people would first look at principal components analysis. So the point of this is to reduce data into a series of principal components, and these explain a decreasing amount of variance. So your first principal component represents the most variance that is explained for the data. And there's an example of that in the figure below on the left. So PC1 here represents the first principal component along the x-axis, and we have the second on the y-axis. And we can see that first principal component explains more than 10% of the actual variance we observe in the data. And these points have been colored because I know which populations some of the samples are from. So all of these yellow ones here are C-lineage bees. All of these black ones are M-lineage. So the biggest variation in the data we observe here is between C and M-lineage bees. And then if we've got the y-axis, we have African bees up at the top, and we have Caucasian bees, a sort of intermediate between African and C-lineage bees. We also have a population of blue bees here, which if you didn't know they were discrete, you'd just assume they were C-lineage bees. So if we look at some of the other principal components, along the third, this separates the Caucasian bees from all of the other bees. And then along this y-axis, we have these blue bees, which are actually from Gotland, which some of you may be aware is an island in the Baltic Sea. They just dumped some bees there, let nature take its course. And after a couple of years, they went down from 150 colonies down to about 9 because they were wiped out largely by Varroa. And then these remaining colonies seem to thrive to some degree. So those populations of bees there are distinct in that sense, and obviously they're of interest because they may have some tolerance to varroa. Another way of looking at that data, as Mark has illustrated, is by an admixture analysis. So this is sort of a genome-wide estimate of ancestry proportions. And this is a global estimate because it's looking at the whole genome that you have data for. This clusters the data similar to the principal component analysis I've just explained. And it determines the optimum number of clusters to explain the data structure. And as Mark was saying, it can do this by first assuming 2 backgrounds and then increasing up to the point where you tell it to stop. So let's say from 2 to 10 backgrounds. It runs all of those models and then it sees which is most likely based on sort of a cross-validation process to see which generates the least error or which simulated data best matches that you've observed. In doing this, we can estimate the ancestry proportions for each cluster for an individual bee. So this large block here on the left titled C is, as you might guess, C-lineage. And we know this because it's mostly Carniolan samples. And again, with M, we know that most of these are mellifera. A is the African, this blue one here is Scotland, and the N is Caucasia. So these are the same bees that the principal component analysis was based on the previous slide. A lot of these M-lineage bees are from places like Colonsay, Wessant, and the Scottish Lothians. Some are from France and Switzerland. The larger admixture samples here are typically those from mainland Europe. Another way of performing this kind of admixture analysis is what's called a supervised analysis. So here we say we have some reference populations. We could take some of those pure bees from the previous slide, representing the Carniolans, the Ligusticans, the black bees, some Africans and some Caucasians, and we can say these are our reference bees. Now here is a query population. Assuming this query population is derived from these reference bees, you can then estimate what its global ancestry values are, which are much like the previous slide, but also along the chromosome in discrete windows. So let's say in 10 kilobase windows, we can say, you know, what background do you observe in that genomic window? And in doing that, we can then look at, are there regions of the genome that are overrepresented? Is there enrichment for a particular genetic background in any of those regions? In this example here, these bees, again, they're Gotland bees, and we know from the history that they're actually buckfast, the majority of them are. So in these buckfast bees, we can see the backgrounds are mainly C with a bit of M and a bit of Caucasian. There is some Africa in there, but it's a tiny, tiny insignificant amount, so it could just be a general background from the ancestry of all bees. And they have enrichment here on chromosome 1. There are a number of immune-related genes in that region. And we've got a paper under review now which covers a lot of these points. So hopefully that will be out soon and you'll be able to take a look at it because it will be open access. So in order to do these kind of reference-based analyses, you need what we call a hat map. And I'll go on to that in a little more detail later. Something else I've been looking at is museum bees. And I've done a study with someone I did my PhD with, well, my postdoc, they were doing their PhD, Melanie Parejo. She studied the Swiss bees because she's interested in the native bees of Switzerland, the black bees. And she was comparing the modern populations to DNA that was sequenced from museum bees. So this is quite tricky because you have to treat the bees as sort of ancient DNA. But nonetheless, she's managed to whole genome sequence these bees. And it lets her see what the differences are between the past and the present, given the time difference between them. So a lot of these bees were from early 20th century, late 19th century. And they're not hugely different, believe it or not. So the agricultural and apicultural revolutions of the late 20th century haven't had a huge impact on bees, not from a diversity point of view. But this is interesting from some context because when it comes to selecting native bees, traditionally this is done on the wing morphometry, so the venation within the wings, or on the hair color. And these traits, they're likely controlled by just a small number of genes, maybe half a dozen or so. Whereas in the actual genome, you've got more than 12,000. So if you're selecting your bees because they look native, you're actually just selecting for a handful of genes within the 12,000 that are present in the genome. So you're selecting for looks rather than local adaptation. Now, the modern way of doing that would be using one of these genotyping assays, like the iPlex mass array that we've mentioned. And this has got SNPs distributed throughout the genome. So you're not focusing on just a few discrete regions. You can select for, or rather across the whole genome using this, this platform. So you don't skew it towards just those 2 or 3 traits that you're looking at. So the museum data that I mentioned is from Switzerland. Post-COVID, when restrictions allow, we'll be able to head into the museums here in Scotland and sample some British Isles pre-1920s bees, sequence those, and that will give us an idea of what the native bee in Britain typically looks like or used to look like. So coming back onto the HAP map, if you're not already aware, honeybee drones are haploid, so they only inherit their alleles from the queen, which means that in theory, they have just a single haplotype essentially for the genome, whereas a diploid like a worker or the queen would have 2 copies of each chromosome, and that's typically called diploid. And although you can identify the variants along the genome, you don't know in what order those variants are. So if you have 3 or 4 genetic variants next to each other, you don't know if it goes ABC or if it's, you know, ACB or BAC. So with drones, we don't have that issue because the haplotypes are what we call phased. So by collecting haplotypes, these drones from all sorts of different genomic backgrounds, we capture the diversity of the species. And if you think from different pressures, selection pressures like climate, pesticides, and so on, this can cause some haplotypes in certain populations to reach fixation because the alleles that they possess might confer an advantage in terms of dealing with those pressures. And we can test for that kind of selection using a number of different methods. And the one example here, it's quite convoluted, but it's called cross-population extended haplotype homozygosity. So in this top plot with the yellow and gray bands, you can see there's a huge peak here on this chromosome. And that peak corresponds with this plot down here beneath it. And this is divided into 5 clusters. And cluster 1 is the population that was being tested, which from the previous slides, you might recall is the C-lineage. And you can see in that population, we have fixation. We have alleles that are fixed in this population because each row represents an individual and each column represents a possible allele for each of those drones. Whereas in the other clusters, you can see, well, apart from these bottom 2, in the M-lineage, for example, you can see the alleles aren't fixed within that cluster. There's a lot of variation happening. This plot to the right of it corresponds to the M-lineage, so the black bees, and you can see there's a peak here on this chromosome, which corresponds again with this plot below it. And we've got selection here in the black bees for that genomic region. So if you were to think in the context of the Roslin bees or any other population that might have some tolerance characteristic to a particular disease, if we have haplotypes from that population, we can compare them against all other populations and find out where those signatures of selection are actually occurring and what genes are involved. So coming back to the reference genome, as I mentioned before, it's linear. It represents a single individual. And when we align sequencing reads to that individual, not all reads will align. Some will come from bacteria, as I mentioned, and these can be assembled and cross-referenced against a database. But there are also differences between different populations, different lineages of bee, which will prevent those reads aligning. So although they're actual honeybee reads, they don't align to that reference genome because there are too many variations in sequence, and they could be structural variations like a deletion, for example. They could be missing that chunk of sequence. They could have an extra sequence inserted. The sequence could be the wrong way round. It could be duplicated if there's a gene duplication. So there's a whole number of reasons why the reads won't align to this linear reference. What we're actually working on now is called a graph genome. So we're incorporating the 2 assemblies that we have. The one I mentioned from the US, the DH4, the INRA Westmont assembly for the M-lineage. I think colleagues also found reasonably good assemblies for a couple of other lineages. Caucasia, I think, will be included in this. But by including these assemblies, what we essentially have is a graph genome which captures the variation from the different lineages. So if you imagine a linear genome is just going from left to right, you either match or you don't. Well, the graph genome, if we start here, you can take a number of different paths to get to the end goal. So each path represents a different type of genetic variant that isn't a single base. So it could be an insertion, or it could be a deletion, an inversion, and so on, which means that by doing this and aligning the reads to that graph genome, more reads align, we capture more genetic diversity, and we can identify structural variants that we would otherwise not be able to identify. And they're quite common, having major impact on how individuals react to certain things. So back onto the IPLEX mass array, as Mark mentioned, this was developed as an inexpensive test of M or C-lineage ancestry. It doesn't just have to be M-lineage. If you hate the little black bees, but you love the little yellow bees, you can equally use it to quantify what the admixture proportions are in the bees. So you can select towards C-lineage or you can select towards M-lineage. Compared the results of an IPLEX run versus whole genome sequencing here. So using whole genome sequence data, which is more than 300,000 SNPs in this case, I've subset it down to the 100 or so on the IPLEX and repeated the analysis. And you can see the clustering, although it's a little bit more scattered, it does separate the yellow bees from the black bees as it intends to, but we also pull out here some bees of African origin. In terms of the correlation between running these two independent analyses, so if you were to do this analysis using whole genome sequence data and repeat it using the IPLEX mass array, the results should give you a very good correlation. It's almost 95% in terms of how consistent the two approaches are. The main difference here being that the IPLEX is considerably cheaper than whole genome sequencing, considerably less intensive in terms of time it takes to process the data, analyze it, and return an admixture estimate. So we know it's a hugely good tool for what it does. Onto the gut microbiome. I know there's been a question on this. So the insect gut microbiome is important. It's important for metabolic function, for stimulating the host immune response, for resistance to pathogens. The taxonomic diversity, so the different bacterial species, is actually quite low, and more than 95% of the gut microbiota is accounted for by just 9 bacterial species, although that can include a number of different strains. Now, when newly emerged bees emerge, hence the term, their gut microbiota is largely missing, and they acquire that through interactions with adult bees, such as nurse bees, and that usually happens within a week. So there's huge potential here from a research point of view to actually test the impact of different compositions of gut microbiota on the health and development of a bee. So from that point of view, there's also potential for diagnostic testing and delivering probiotic treatments. So concoctions essentially of friendly bacteria for bees that could help with dealing with things like pathogens or tricky environments. So if you have your bees in an area where the landscape is quite poor in forage, poor in pollen, then it could be quite useful to have that kind of supplement. There's also, in the long distant future, when the public aren't so afraid of it, potential for genetic engineering of the gut symbionts to confer resistance to pathogens. And it can do this. So if you take, it's been demonstrated, if you take one of the species, for example, Snodgrassella, you can genetically engineer that to produce double-stranded RNA, which then triggers a response in the host and varroa, reducing the mortality rate in the bees. So it reduces the effect of varroa in terms of how successful it is and the amount of virus that causes problems for the bee. And a similar thing's also been done looking at Nosema. Finally, I believe, meta-barcoding may be of interest. So this is where we extract DNA either from pollen or from the guts of the bees. And where you look to sequence just a tiny, tiny stretch of the DNA, which is sufficient to identify species to a taxonomic degree. So it's not species level in all cases, but it can be genus level. And what that tells us is the diversity of species either within the bee's gut or within the bee's diet. And this is much like whole genome sequencing in the sense that we extract the DNA, we amplify up that short region that we're interested in, we sequence that, and that generates millions of reads. And we can tell from those millions of reads which species are present and how abundant they are. And these reads are referenced against databases that are publicly available. So that can be quite useful from a health diagnostics point of view, and also from a landscape diet point of view. And that's me. So thank you very much for listening.
So Mark and David have talked about all the fantastic work that's going on at the moment with genetics and genomics, and all the research that's happening, and all the new tools and new facilities that are available for doing all this work. And I think for a lot of people, it's fascinating, but it's also quite overwhelming. And we certainly discovered this as beekeepers, talking to people, and people say, well, this all sounds really fascinating. How do I get my hands on this? And, you know, how is it going to work, and how much does it cost, and who do I speak to? And this is really where the idea for Beebytes came about, was realizing that there's an interface that needs to happen between the people like Mark and David who are working at the cutting edge of research, creating these new tests and new tools, and the beekeepers who want to make use of these, but don't necessarily have the scientific background or the access to these resources. So Beebytes is a community interest company. It's a social enterprise, and it's a not-for-profit. So we don't have any shareholders, we don't pay any dividends, and any profit that's made by the company is to be used to benefit the community. So ultimately, the Beebytes is self-sustaining, and aims to reuse any funds that it raises in order to help improve all aspects of bee health and bee genetic research. We're based at the Roslin Innovation Centre, which is directly next door to the Roslin Institute, and is managed and run by the University of Edinburgh. And we have had a lot of support from them in getting set up. The Innovation Centre is particularly focused on insect research at the moment. There are a lot of companies, lots of spin-outs that are doing work with insects. So it's very good for us to be in that location, because there's a huge number of people working both in research and commercially in the field of insect research. So it's a good place for us to be in terms of having access to expertise as we develop our company. Between the three of us, we have a great amount of expertise in this area. Obviously, you've heard from Mark and David talking about the honeybee genomics, bioinformatics, statistical genetics. Mark and myself are both Scottish Expert Beemasters. We've been keeping bees for a good number of years, doing teaching and research work. So we understand the beekeeping side of things. And my personal background is in large-scale data analysis and software engineering. So in terms of doing the data analysis and making that available and understandable to people. So we think that we cross the board in terms of everything from the genomic side of things right through beekeeping, right through to the data presentation on the other side. So our core aims for Beebytes, probably the top one as a company is low-cost services. We're looking to provide easy access to genetic testing and all the related services that go along with this. So all of the things that David and Mark have talked about, if there's a way that we can implement that as a service that's available to beekeepers, bee breeders, people working in any environmental research, that's our aim, is to try and make that available in an easy to access, low cost way. Obviously our main focus is on honeybees and beekeeping, but these things allow us to test. We've talked about pollen, meta-barcoding, microbiome. We could be looking at testing pollen in an area for environmental surveys for biodiversity, or looking at the microbiome present in the walls of a colony and comparing, say, the hive lining to that of wild hives. There's lots of areas we could be doing genetic testing, and so we're open to any ideas about what people might want in terms of services. Another one of our core aims is open data. The idea behind the open data movement is that any data that's being produced by science should be freely available to everyone to use. It shouldn't be encumbered with patents or copyright, trademarks, things like that. So any data that we collect and gather, we want to make that available for people to use, whether that's individual beekeepers wanting to find out more about the bees in their area, or other people working in research in these fields. We don't want to make it so that the data is all tied up and owned by the company and you have to pay lots of money to access it. That's completely against the ideals that we're aiming for here. And finally, as a social enterprise, we're all about reinvestment. So any profit that comes from offering these services will go into research and development, developing new tests, new services, reducing the cost and increasing the availability. We hope to be able to do more outreach, more education work, working with schools, science festivals, universities, these kind of locations to get people more involved in bee research and pollinators, and just generally promote pollinator health and biodiversity. This is an area that's very important to us. So to begin with, we're effectively starting with a single product. We hope to expand that range, but to begin with, we're going along the adage of, do one thing and do it well. The first product we're offering is this MC-lineage admixture assay, which is comparing the levels of hybridization or admixture in colonies. So effectively, it's looking at M-lineage versus C-lineage in colonies. We know this is very important to a number of people. There are many conservation groups looking at the native black bee in Britain and in Europe. There are also lots of bee breeders, particularly breeding strains such as Ligustica or Buckfast, who would be interested in this kind of data. So that's the place that we're starting our work. We're basing it on the IPLEX mass array. As David explained, that gives us pretty much nearly the same accuracy, 95% accuracy as whole genome sequencing, but at a much, much lower cost and with much faster turnaround. In terms of actually sampling, we need a sample of 15 individual drone antennae. So that's a single antenna from 15 drones from a single colony. And that allows us to then analyze the genetics in those drones and effectively, like a jigsaw, rebuild the genetics of the queen that produced those drones. And then that gives us the idea of the hybridization and admixture levels in that queen. For that test, the cost is £59, and we expect to have a 3 to 4 week turnaround on that. So we hope that people will be able to be sampling drones early in the season. We should be able to give them a response quickly, and they should be able to then use that information to change what they do in terms of their breeding programs or their selection of lineages, all in the same season. Whereas we know with previous genetic tests, it's often been a case that samples may have taken several months, and there's a problem there to select and make decisions when you're having to wait till the next season to be able to do it. In terms of sampling the drones, we sample from pupae. So the reason for doing that is that you end up with the guaranteeing that the drones from that colony, drones naturally travel between colonies as adults. So we don't sample on adult drones because there's a risk that those drones have flown in from other colonies. And the reason for sampling drones and looking at the queen lineage rather than sampling workers is that there's been a lot of recent research that shows that the polyandry of queens may be much, much larger than we expect. Queens may mate with as many as 40 or more drones in their mating flight, and it's impossible to sample enough workers to accurately reflect the genetics of the queen and all the drones she mated with. So by sampling the drones, we can guarantee that because their genetics only reflect that of the queen, we can guarantee the lineage of that queen. So the process for sampling is fairly straightforward. An order is placed through the website. We dispatch a DNA collection kit with instructions, which in the case of drone sampling is really some sample tubes with a extraction buffer and some tweezers. The beekeeper collects the drone antennae, pops them in the tube, sticks them back in the post to us. We then take the DNA, do the extraction work, get the processing done in the lab. Once the raw data is available to us, we then do another level of analysis on top of that and produce the results in a nice, easy to consume format for the beekeeper. And then the data from those results then gets added to the collection of data that we've got and made freely available through our open data model. In terms of open data, we hope that we'll have all the results available to everyone. That's everything from raw data through to nicely formatted data in terms of graphs and interactive mapping, things like that. Obviously, the results will be anonymized. There'll be no information about the beekeeper involved. So every sample will be associated with a beekeeper, but only the beekeeper and ourselves will know that. The information that's published will be anonymized. and we'll be anonymizing the data as well over grid cells, probably 10-kilometer areas, in the same way as Beebase does, so that there's no information about the exact location of hives for people who have concern about hive security. Once we start collecting different samples, we'll be able to provide data in many different data sets. So obviously we're starting with honeybee hybridization, but we could be looking at pollen availability, pollen diversity, different times of year, different seasons. We could be looking at chemical analysis. We could look at whole genome sequencing and microbiome. But over time, we hope to build a huge data set that will allow everyone from beekeepers to researchers to get an idea about what's available in their area and what they can be doing to help improve the bee health and the quality of the bees that they keep. So thank you very much for listening to us this evening. Put the link there to the website. We're not currently, unfortunately, selling products. We hope that we will be going live in May. We're just in the final steps now of finalizing everything. Annoyingly, mostly it's administrative and legal things at this point. But feel free to get in touch with us, visit our website, and contact us by email if you have any questions about our services. And I'll stop sharing now, and we can open up for any general questions to the three of us about anything we've talked about this evening.
Brilliant. Thank you, Matthew. There are some questions which have come in via the chat function, which I'm sure you can see as well. Would one of you care to address one of those rather than me read them out, or shall I read them out? Hello, is anybody there?
Sorry, I'm just reading them on their own.
Yes. So we've got one introductory text say that queens mate several times to reduce the risk of CSD gene locus delivering a diploid drone. Do other honeybee genes behave in a similar way? So
with regards to the CSD locus, this is one of the mechanisms for having, male and female bees, along with another called a feminizer, I believe. So the CSD locus it's hypervariable. So the queen will mate with multiple drones, typically more than 12. And in theory, those drones will hopefully have a different CSD locus, so that the queen's offspring, when they combine her CSD with those of the drones she's mated with, won't have the same CSD allele. Now, in the unlikely event that that does happen, you end up with a diploid drone, and obviously the colony rejects it. As far as I'm aware, that's the only gene that behaves in that manner in the honeybee.
Thank you, David. I had a question. When the Gotland bees, I didn't realize that they were still being studied. That study where they put 150 colonies was many, many years ago now. I'm really quite surprised that was going on. Did they ever get to the bottom of whether it was the bees becoming resistant to Varroa, or the Varroa were becoming less aggressive towards the bees?
I don't think it's been resolved. I heard from a little birdie that in order to protect these surviving colonies, that people started treating them, which intuitively, is against the point.
Yeah.
But that sequence data was generated by another group. We've essentially taken publicly available data as much as we could get. I think there was about 689 individual drones that we processed in the end. Yeah. And that was part of a project funded by the CB Dennis Trust. Yeah. But what situations with the bees on that island now, I'm not sure.
I always appear to be the case that the bees there have undergone enough genetic variation over the time they've been there for them to show up as distinct in the population when compared to a sort of default background of buckfast bees. And as David was saying, there seems to be some extra expression in areas that are believed to be associated with immune response. But whether that's a direct response in some way that proactively helps against Varroa, or whether there's some other factor that's caused that variation, isn't clear.
Yes. I thought it was a nice example of be careful what you wish for, because as I understood it, the colonies, although they had resistance to Varroa, were small, non-productive, swarmed at the drop of a hat and fierce as hell. So not quite the sort of ideal bee colony.
Bob, I could answer Keith Hooker's question, I think, on whether you can identify viral loads. Yes, you can. It's probably just a little bit expensive, or quite a lot of work at the moment. You could use RNA sequencing to do that, or you could use basically you can do PCR from detect RNA by RT-PCR, but it's not very easy to make that into a sort of the kind of service we could do at the moment, but it's certainly something we'd aspire to in the future.
Could I ask whether you're applying some of these tools to your university colonies, and what where you're trying to go with them?
We've, I mean, the apiaries we've got are rather quite small. You know, my Easter bush is about 6 colonies generally. We generally use it for getting material for things like the atlas, etc. I know David does have some plans for an experiment, but regarding resistance to a certain disease. So I guess the answer is yes in the future.
Okay. Are there any other questions? Is there anyone listening, you know, live, as it were, who would like to unmute and pose a question? Bit of a silence there. Okay, I've got one more. I noticed, Mark, when you were talking about the bacteria and the fungal things that you were able to spot, that you saw Ascophera apis chalk brood. I wondered whether, I mean, stone brood keeps cropping up as something which I've been keeping bees for a long time. I don't think I've ever seen stone brood, and I just wonder whether it's actually still around.
Yeah, I mean that experiment was only based on 19 colonies. So
okay.
Also it depends whether it's in the databases as well. You know, stone brood has to be in the databases for us to find it.
Mm.
But that would be, yeah, we should have a look and see if it is.
Okay. Right. Well, we're almost exactly 60 minutes. And if there aren't any further questions, it only remains for me to thank you very, very much. Thank you to Mark, to David, and to Matthew. Obviously working very hard to understand and to provide this service for UK beekeepers. So all power to your elbow. I hope that this works out well. And once more, thank you very much indeed.